So this last part here, we're going to talk about some more things that can happen that don't exactly follow those dominant or recessive rules that we talked about with alleles. So right here, the next thing we're going to talk about is going to be something called codominance. And codominance is going to be different from incomplete dominance. In incomplete dominance, what happens there is you have an intermediate version showing up. For example, the example I gave you with the flower. So you've got a red flower crossed with a white flower, and you get a pink flower, which is just kind of in between. So neither of them are fully showing up. They're just kind of showing up halfway. That's why it's called incomplete dominance. Now, the next part that we're going to talk about that is codominance is going to be with the ABO blood group. Now, A and B are going to be dominant, and O is going to be recessive. Okay? So if we try and think about the different blood types that are out there, let me just scroll through here a little bit. Okay, our first blood type is going to be type A. Then we've got type B, type O, and then type AB blood. Okay? So keep in mind that A is dominant, B is dominant, and O is recessive. Okay? So keep that in mind. Now, if we talk about someone who has type A blood, there are two possibilities for their genotype. They can either be AA or AO, right? Because O is recessive. If you have type B blood, think about that. That means you're either going to be BB or everyone's favorite BO, right? Because O is recessive. If you have type O blood, then that means there's only one combination you can have. Remember, O is recessive, so you would have to be OO. And if you have type AB blood, that means you're going to have one of each, A and B. So that AB blood is going to be the example of codominance. Now, as far as what this looks like phenotypically, so you're going to have plasma, and then you're going to have your red blood cells. Now, your red blood cells are going to have receptors on them. You can see the little pink things going around here. And then your plasma is going to have antibodies in it. Okay? So these little receptors on the outside of the red blood cells, those are called antigens. So the key is you never want the antigens and the antibodies to link up with each other and match, because if they do, then you're going to end up with blood clots. So someone who has type A blood is going to have A antigens, anti-B antibodies, and that's okay because the pink and the green are never going to actually match up. Okay? Now, if you have type B blood, you're going to have type B antigens on here, and then anti-A antibodies in the plasma. If you have type AB blood, you're obviously going to have both A and B antigens. You're going to have no antibodies present. And then if you have type O blood, you're going to have no antigens on your cells. And in your plasma, you're going to have both anti-A and anti-B antibodies. Now, how does this work as far as donations go? Well, when you donate blood, what they're really going to be giving you is those red blood cells. So let's say you have type A blood and someone with type B blood donates to you, okay? So you're getting a bunch of these mixing with this whole setup here. Obviously, this is not going to turn out well because these right here are going to match up with this right here. And so you're going to have clots, and clots are not going to be a good thing, and you could die. Same thing is if you have one who has type B blood and you give them A. So you have these guys that are going to match up with this, and that's going to cause clots, and then you could die from that. Now, someone who has type AB blood can receive either A, B, or AB because, and actually O as well, because it doesn't have any antibodies present. So we say that AB is the universal recipient. It can take any of these because it doesn't have those antibodies present. Now, if someone has type O blood, we call them the universal donor. Now think about why they would be the universal donor. Well, from O to AB, no big deal because there's no antibodies in AB, and we said it's the universal recipient. For O to go into B, it doesn't have any antigens on the outside, so it doesn't matter about these right here. And the same thing goes with here. So that's why O is going to be the universal donor. Now O can only receive O, and the reason for that is because of this right here. It has both anti-A and anti-B antigen or antibodies present. So any of these could match up with these, so you can only receive O if you have O. Okay? So that's how those blood types work. Now, a question you might be asking yourself is, what about the positive and negative part? That's going to be called the RH group. And the way RH groups work is that RH positive is going to be dominant and negative is going to be recessive. And the only time that's really a big deal is when you talk about childbirth, because if you are a person who is Rh negative and the baby's blood is Rh positive, the immune system will attack and you can have major complications. And that's honestly what they think was a big problem with childbirth and a lot of the deaths that happened in childbirth long ago. So now it's not a big deal. They can actually give you an injection and infuse you and you're fine. But that is something that they want to make sure they know about so that they can prepare for that. Now this next part is talking about pedigrees. And pedigrees are a way of looking at genetic traits over time to see how something kind of flows through a family. So in this next slide here, this is a basic key to a pedigree. So anytime you have a horizontal line between a square and a circle, that's going to say that you've got a mated pair. So they have some type of offspring. And then the offspring are going to be in a line down from them. And males are going to be squares, females are going to be circles. If they have the homozygous recessive trait, they're usually going to be filled in, but they don't have to be filled in all the time. Now, if you look at this pedigree here, you can see the only ones that are filled in with color are going to be the homozygous recessive people in that family. And so you can see here, this is a couple who were both heterozygous, and they had offspring. This one, this one, this one, and this one are their offspring. This one married this one, but this one came from these parents up here, okay? So that's how you can look at a pedigree. Now, if we look at this one here, this is a pedigree showing how hemophilia actually was passed through the British royal family back in the day. And the way that we use this is, once again, males are square, females are circle, and if you're affected, you're going to be colored all the way in. If you're a carrier, you're only going to be colored in halfway. So take a look at this and tell me if you notice anything weird about who gets affected by hemophilia. If you look, you can see that only the squares are the ones that are filled in with green. So only males are showing to be affected by this. The females are carriers. Now, is it possible for a female to get it? Sure. But really, it's the males that tend to get it much more often. And that has to do with something we're going to talk about in a second. Now, something I could ask you on a test is I could show you this. I wouldn't have this key up here, but I could say, tell me about person number 4. And what you could tell me was that, well, person number 4 is a normal male. If I said, tell me about Alice, you could say, well, Alice is a female who's a carrier for that trait. So that would be something I could ask you about these different things. All right, let's go back to your notes. So hemophilia is what I was just showing you there, and that's going to be something called an X-linked allele, and I'm going to actually go over that with you in class, and I'm also going to attach a video that'll talk about that more. Oops, sorry about that. Okay. So genetic recombination, we've talked about that in meiosis. That's going to be where you have chromosomes and the crossing over is happening. And with that, you can use that to make what's called a genetic map. And a genetic map is going to be this right here. So this is actually what they used for the Human Genome Project. And so they use those sites where crossing over happened to kind of map out where those genes are actually located on a chromosome. And so this is chromosome X, and this is only showing bad stuff, but this is showing things that could go wrong on chromosome X and where you would find it. The reason we think this is cool is because we could actually, let's say we have someone who has retinitis pigmentosa, we could actually extract the bad part of that gene and insert a healthy version, and now we've done gene therapy. So that's why that's really important. Okay, now we've talked about human chromosomes. Now we have 23 pairs. 22 of them are not sex cells, and so those are going to be called autosomes. And the remaining pair is called the sex chromosomes. And females are going to be XX, and males are going to be XY. So technically, we all start out the same, and then that Y chromosome would be what would trigger a male to create secondary male characteristics. Um, so if it doesn't have that Y chromosome, then the individual is just going to remain a female. Now, a couple of things that can go down with chromosome number that we've talked about. Down syndrome, that's when you have an extra chromosome 21, and that can cause some genetic defects to show up. I thought I had an example. Yeah, here. So in this karyotype, you can see right here, there's the problem, right? You have 3 chromosomes there where you should only have 2 for chromosome 21. So that's an example of trisomy or Down syndrome. Now, another thing we can talk about is sex-linked genes, and that's what hemophilia was an example of. And like I said, I'm going to post an example of that online for you guys, and I'm also going to go over that with you in class. Then the last thing we're going to talk about is going to be inactivation in females. So we've talked about the fact that females have 2 X chromosomes, and so if you have 2 X's, you don't really need that second one. And so what can happen is you can actually have this occur where these 2 blobs are going to be chromosomes, and in a male you can see the X and the Y are both pretty clearly represented. But in a female, since it doesn't need that second X, it has that one that you can see, and then the other one kind of fuses and eventually becomes part of the nuclear membrane, and we call those bar bodies. So just by looking at the nucleus of a cell, you can sometimes tell whether it's male or female by looking for those bar bodies to be incorporated in the side of the nuclear envelope. So that's it for Chapter 14.